asrafil anbiya'i wal mursalin wa ala alihi wa sahbihi ajma'in amma ba'du. Very good morning, ladies and gentlemen, the honorable speakers, Dr. Juliana binti Jumal from University Science Islam Malaysia, the honorable speaker, Dr. Tatang Julianto from Chemistry Department, Universitas Islam Indonesia, our world-class professor, Professor Wi Antung from National Tsinghua University. It is very happy to have all of you in this screen for this morning. We will have the webinar series of Chemistry Department, Universitas Islam Indonesia. And this morning, we will invite the topic of organic chemistry for future life. So let me start the screen for today. Wait.
Prof, it seems that the line is still not stable.
I guess it's okay. Okay, so before we start this event, it is kindly requested to all participants to rename the Zoom account with your name underscores institution for us to showcase your identity. Switch off your microphone during the session unless the moderator or host welcome you to ask question by direct speaking. And as you can replace your background, please use the virtual background of the event so we will have nice photo for our documentation after welcoming remark. And if you have question, please write in the chat, which will be open in 15 minutes before the speech from the speaker finish. And in case you have trouble in signing in the Zoom meeting, you can broadcast the webinar on YouTube channel, Jurusan Kimia FMIPA UII. So Bapak Ibu, supaya agenda ini berjalan dengan lancar, pertama kami mohon untuk mengubah atau merename akun Bapak Ibu dengan nama underscore institusi. Kemudian dimohon bagi Bapak Ibu untuk mematikan mikrofon selama sesi, kecuali nanti ketika moderator atau host meminta Bapak Ibu untuk bertanya. Kemudian bagi Bapak Ibu yang dapat mengganti virtual background, silakan mengganti dengan virtual background yang sudah kami kirimkan melalui email. Dan apabila selama diskusi nanti Bapak Ibu memiliki pertanyaan, silakan disampaikan pertanyaannya melalui room chat. Nah, nanti barangkali karena alasan internet dan sebagainya, seandainya Bapak Ibu mengalami kesulitan di dalam Zoom ini, Bapak Ibu bisa melakukan mengikuti webinar ini di channel YouTube ya Jurusan Kimia FMIPA UI. Sekali lagi dimohon bagi Bapak Ibu peserta untuk mematikan mikrofon. Okay, so these are the rundown of this meeting. We will have the welcoming remark from Prof. Wi Antung first, before the speech from Dr. Juliana binti Jumal, followed by the speech from Dr. Tatang Julianto, and the agenda will be finished at around 12 p.m. Okay, so ladies and gentlemen, as we know that the development of industrial technology cannot be separated with the chemistry. Moreover, in this pandemic situation, everyone needs to stay healthy with high immunity. So vitamin, medicine, and of course vaccine are the important thing. And the very foundation of biochemistry, biotechnology, and medicinal are built on organic chemistry and its role in life process. Furthermore, the high technology of materials are at least composed of organic chemistry together with other branches of chemistry. So with this webinar, we will hear and discuss of the sharing from the person who is closely conducted many research progress in studying and developing organic chemistry research. We will have Dr. Juliana binti Jumal from University Sains Islam Malaysia, and we will also have Dr. Tatang Sabur Julianto from Chemistry Department Universitas Islam Indonesia. And okay, well, as welcoming remark, we will hear the important point remark from Prof. Rui Andong from National Tsinghua University. Okay, Prof, time is yours.
Okay, thank you for the invitation. Good morning, everyone. Also, good morning, Professor is Fatma and our two speakers, Professor Juliana Jomas and Professor Tatang Juliantos. I'm Riandong, Dean of the Tsinghua International College, and also chairs the Professor of Institute of Analytical and Environmental Science, Tsinghua University. Actually, I also studied world-class professors of UII, so that's one of the reasons I'm here to give the opening remarks. Today is my great pleasure to give these opportunities to welcome all of you to attend this webinar, and also have the chance to give some words to you. But on behalf of the world-class professors of UII, I would like us to cordially welcome you to attend this meaningful webinar seminar. And before some my words, I would like us just to thank the UIIs to give these opportunities to me, and I would also like us to congratulate the Department of Chemistry UII for this success conference. I know that to get this funding is very difficult, and when they get this funding, the UII, especially Professor Ismail, he try hard to organize a lot of conference. And one conference I attended before is the topics for energy and environment. During that case, I attended the 3 conference names. I know that the more than 800 people has attended that conference, and everybody really enjoys the atmospheres and the contents that they provide. And the finish of the webinars series 1 names UII has opened another webinar series. This webinars that they focus on organics chemistry for future life. Actually, this is quite the good topics. My understanding is that the extractions of secondaries metabolized from plant is very important worldwide, and especially in Asian country, because in Asian country we located in tropical and subtropical area. We produce a lot of different kinds of species of plant, and for those plants, they contain the different kinds of the function. For example, some functions can cure the cancer, and some chemicals can do the essential oils for the mass therapy. So that's the topic here. Actually, is very important for us. They are not just only provides the fundamental ideas, also provides the big opportunities to commercialize, to market. So I believe that the lowest extracts and metabolized from plants that introduced today will give you a good ideas to use names in a wide application and impact to our daily life. For example, the essential oil from plants and anti-cancer drugs that are from plants. Also, we also can produce some nutrients from fermented products and cosmetic ingredients. That will be the good ways for us to learn the fundamental as well as advance the knowledge from this webinar. So I also believe that this webinars will serve as the platforms to introduce, use the good knowledge is on extraction as well as on purification of secondary metabolites from PREM. I also still believe that all the attendees will learn a lot of the cutting-edge technologies in this field. Last but not least, I believe that these webinars will help us to establish the cross-academic cooperation between UII and Southeast Asian country. Definitely, this online conference will be very successful. I also believe everyone in this conference will enjoy the atmosphere provided by UIIs. I hope that it will have the great times that during the conference and learn something special today. Thank you and enjoy this webinar. Thank you.
Okay, thank you very much, Prof. Rui, for very interesting and inspiring remark. So, ladies and gentlemen, before we start this webinar and enjoy the speech from two speakers, it is kindly requested to all of you to switch your video on so the admin can shoot your screen for our documentation. Bagi Bapak Ibu, untuk dokumentasi kami persilakan untuk menyalakan video agar admin dapat men-screenshot layar untuk kami dokumentasikan. Mari silahkan. So please give your best smile on screen. So we will have very nice documentation. And it is to inform you, Prof. Rui, that we have about 400 registered participants coming from around the world, mainly from Indonesia, but there are also coming from Pakistan, Bangladesh, India, and usually because of limitation of Zoom meeting, sometimes they also follow in YouTube channel. Okay. Smile. Have you finished, Mas Admin?
Yes, finish.
Okay, thank you very much for your smile on screen. So we will start this main agenda for today. We will have Miss Lina Fauzia as the moderator. Good morning, Miss Lina.
Good morning, Prof.
Yeah, okay, so start the speech.
Okay, the Honorable Mr. and Madam Speakers, Professor Juliana and Dr. Tatang Sabur Julianto, the Honorable, the host, Professor Is Fatima and Professor Yandong, distinguished guests, and all of the participants in today's webinar. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Waalaikumsalam warahmatullahi wabarakatuh.
Thanks to Allah that give us healthy and time so we can have discussion today. Once again, welcome to Chemistry Department Universitas Islam Indonesia webinar series, sponsored by World Class Professor. My name is Lina Fauzia from Chemistry Department Universitas Islam Indonesia, and I would like to thank Professor Is Fatima and Professor Riandong that give me opportunity to join this webinar as moderator. Okay, just a little housekeeping before we get started. We will have two speakers, amazing speakers, and 30 minutes for each speaker to deliver the presentation and followed by Q&A session. So if you have any question during the presentation, please type them into the chat box in your Zoom or in YouTube. I will bring them up during the presentation. Okay. May I share the screen?
Yeah.
Okay. Talking about organic chemistry, we are all ring. Why is organic chemistry considered difficult, whereas it has a strong connection with our daily life? Do you hear me? I'm sorry.
Okay.
Yes,
Okay,
yes, we
may I
hear you.
start? Okay, all right. Okay, thank you to all, especially to Prof. Fatima for inviting me. Okay, my name is Juliana. So this is actually one of the part of the research from one of my students on the compounds of 6-hydroxy 4-methyl-coumarin derivatives and their complexes. And we tried to have these compounds to be tested as potential anti-cancer agent. So before we start with what we call as organic synthesis, let's start with what is actually organic chemistry. Okay, we know that organic chemistry plays an important part in our daily life. We can see food, clothes, paper, ink, rubber, soap, perfumes, medicine, and so forth are indispensable for us, to us for proper living. Therefore, we can say that mainly all organic compounds are important constituents of many products, and all known life is based on these organic compounds. That's why we can see paint, food, plastic, explosives, medicine, petrochemical, and pesticide, and so on and so forth are all actually organic compounds. So the importance of study organic chemistry is especially to chemists as us and also pharmacists to synthesize the medicine to the elevation of human suffering. Therefore, this is actually our main expert on organic synthesis, because this organic synthesis have the power to replicate some of the most intriguing molecules of living nature in the laboratory, apply their developed synthetic strategies and technologies to construct various of them, and such molecules facilitate biology and medicine, as they often find uses as biological tools and drug candidates for clinical development. And thus, we can say that by employing sophisticated catalytic reaction, appropriately designed synthetic processes to synthesize not only the molecule nature and also the molecules, but also myriad other organic molecules for the applications in many areas of science, technologies, and everyday life. And you know that synthesis is of paramount importance to our well-being, for through it, we create new chemical entities such as molecules, from which we derive our most precious material items. And we know that organic synthesis have led to an impressive benefit to society, including useful products ranging from pharmaceuticals, dyes, cosmetics, agricultural chemicals, etc. Okay, so this study or this speech is actually highlights on the synthesis of coumarin derivatives and the anti-cancer screening activity on human life. Okay, so this is actually the topic. We already synthesized a newly 6-hydroxy 4-methyl-coumarin derivatives together with the complexes. And actually we focus on these two metals, which is copper and also cobalt. And we tested it for the screening activity, anti-cancer screening activity on this anti-cancer agent. Okay, as we know, cancer is actually a serious and life-threatening disease that causes significant harm to human health. It seems that WHO estimated in 2030 more than 13 million people dying due to cancers. So over this fact, anti-cancer agent is very important in the prevention of the disease. Many different mechanisms how anti-cancer drugs can inhibit the division of cancer cells as the NA intercalating agent, cross-linking agent, etc. However, most of the drugs also severely affect the non-cancerous tissues. So due to that particular problems of cytotoxicity and drug resistance with current chemotherapy agents, the discovery and the development of effective anti-cancer drugs will remain a great challenge. So in pursuit of meeting the challenge, researchers identified numerous compounds having inhibitory activities against various types of cancers with fewer side effects. Okay, my first question, why is coumarin? Okay, basically this is actually the structure of coumarin. It's a fused benzene and pyrrone ring system. Okay, as you can see from the structure of this coumarin, is also called as an heterocyclic compound, and we know that
Dr. Juliana, you are still in mute mode.
Professor Juliana?
okay,
Yes, already on. Yes.
Okay, because I can see the signal, the internet is not stable. Sorry for that. Okay, okay. Extremely coumarin is actually a variable in structure due to the various types of substitutions. Because you can see here the substitutions of this coumarin, we have R3, R4 until R8. So due to that various types of substitutions, we can synthesize various derivatives. Okay, so one of the various derivatives of coumarin is hydroxycoumarin, meaning that the OH group is attached to one of the substituents. So we call this as hydroxycoumarin. It's a typical phenolic compound, metal chelators in complex formation. So instead of the ligand itself, we try to synthesize complex formation, and this hydroxycoumarin is actually a very potent metal chelators in complex formation. And also coumarin derivatives complexes, it seems that can possess immeasurable anti-cancer and have a very potential with minimum effect. Okay, next. Okay, now anti-cancer activity of coumarin derivatives. So this is actually the previous, what we call the literature reviews that comes from researchers since 2012. It seems that some of the coumarin derivatives and the complexes, for example, from Thomas et al., also tried to have this tested on this anti-cancer activity. We also have this 4,4-dipyridine ethoxybenzyl 7-hydroxy. We also have 3-oxopropyl 2-chromine-2-ene. We also have chromine 2,4-dione from Usman 2017. We also have complex with coumarin with nanstrolate from Zhu et al. 2014. We also have copper-2 complexes shift ligand catalase, coumarin shift ligand shift bases, and we also have this all the complexes of 3,1-amino thiolidine 2-chromine 2-dione from Siwatik and Kufel Neke that also study on these coumarin derivatives. And we can see from all these researches, the metal complexes tend to be more potent as anti-cancer agent compared to ligand. And due to that, it's actually because of the different mechanism activity will provide alternative route of novel drugs and the ability to bind and cleavage the MA, the species, what we call as ROS or reactive oxygen species, being generated, which actually induce the cell death. This is actually part of the literature review from what we call these coumarin derivatives. So the objective of our study first is to synthesize bispecific on this compound, 6-hydroxy-4-methyl-coumarin, and also the derivatives together with the complexes. And we focus on these two metals of copper and also cobalt. After we synthesize the compound, we characterize the synthesized compounds by using CHN elemental analysis and also the spectroscopy methods of FTIR and proton NMR. And finally, after we already successfully synthesize and characterize, we try to study the anti-cancer activity of the synthesized compounds by using MTT assay against this, what we call a cell MCF7 cell. And we will try to focus more on this cell. Actually, it's a cancer lung cell. Okay, so how the methodology or the research methodology? Actually, it covered 3 stages. Okay, the first stage is actually the synthesis. So basically, we already synthesized 10 compounds. C1 is actually the first ligand, which is 6-hydroxy-4-methyl-coumarin. Then we proceed to the azo-coumarin. We call it as C2, C3, C4, where this halo-phenyl-azo-coumarin, we just specify on this halogen of chloro, bromo, and nitro. And then we proceed to the C5, C6, C7, C8, C8, 9, and 10, which are metal complexes of 6-hydroxyl-4-methyl-5,7 bis para X, which is actually the halogen, phenyl-azo-coumarin, where the M is actually copper and also cobalt. Next, we'll go to the characterization, as I already stated just now, the CHN analysis, the FTIR, and also the proton NMR spectroscopy methods. And the last stage will be the anti-cancer screen activity. Okay, we actually use MTT assay, where the cell is MCF7 cells, and then we use the ELISA reader to find the optical density or OD. From this ELISA reader at a certain wavelength, we try to get the percentage cell viability and also the IC50 value. So this is basically the 3 stages actually covered for the method. Okay. For the results and the discussion first when it comes to synthesis. Okay, so what are the starting materials? So these are the starting material. We have hydroquinone, reactive ethyl acetate to get your first product or the ligand, okay, C1, which is 6-hydroxy-4-methyl-coumarin. And then from that, next we'll try to synthesize the azo-coumarin, where we have para-haloaniline. We react with sodium nitrite together with strong acid in the presence of ethanol at very low temperature, about -10 degrees Celsius, and that would be your azo-coumarin. Okay, after that, when you have your azo-coumarin, okay, we get our first C1, which is 6-hydroxy-4-methyl-coumarin, react with the azo-coumarin and strong base sodium hydroxide to produce the ligand of 6-hydroxy-4-methyl-halophenyl-azo-coumarin, and we focus on the X, which is chloro, bromo, and nitro. Okay, step 3 is where we have this. Since we can see from the previous literature review, so it shows that the complex actually tends to have more potent anti-cancer reactions. So that's why we try to have to synthesize the metal complex, where we focus on the metal of copper and also cobalt, and that will be the product of C5, C6, C7, C8, C9, and C10, which are all the metal complex of the 6-hydroxy-4-methyl coumarin derivatives. Okay, so basically there are 10 coumarin derivatives that are already being synthesized. So this is actually the summary where we have your step 1, the ligand, the first ligand, step 2, the azo-methyl ligand, and step 3 is the metal complex. Okay, so these are the 10 products or the 10 compounds that are already been synthesized regarding the physical properties. So the color we can see, we have various colors from the C1 compounds from pale yellowish until we finally try to synthesize to produce C10, which is actually the metal cobalt, where we have brownish purple. Okay, all right. Okay, next we have, this is the molecular formula of the compound of C1 until C5, where you can see the melting point is more than 100 degrees, but when it comes to the metal complex, it becomes higher, about more than 200 degrees. And the CH analysis is to show the percentage of the elemental of carbon and hydrogen, and the asterisk is to show the theoretical value. And from the value, it shows that not that much difference between the experimental and also the theoretical value of the CHN analysis. Okay, next we'll go to compound C6 until C10. All these are the metals.
Please unmute, Doctor.
The line, I think the internet line is actually automatic, it's unmuted. Can you hear me?
Yes, yes.
Okay, all right. So this is actually the compounds. So C6 until C10, we show the percentage of the elements of carbon and hydrogen. And it seems there's not that much difference of the experimental and the theoretical value. Okay, so this is the first part of the characterization, where you can see the physical properties, the color, and also the melting point. Okay, next we'll go to the FTIR characterization. Okay, we can see here from C1 until C10, these are the FTIR frequency and the band assignments of the compounds. Okay, so this is actually summary. And from the summary, we can see the band assignment of all the important functional groups. And we can see the important functional groups of your OH, of the CH aromatic ring, the C double bond O, the C double bond C aromatic ring, and actually goes to the CH, CO, CN, and N double bond N if it's actually involved the azo coumarin derivatives. Okay, so I choose out of 10. This is actually to represent the FTIR spectrum of compound C1, C2, and C8. Okay, C1 is the first ligand, which is 6-hydroxy-4-methyl coumarin. And if you look at the spectrum here, we can see a very broad band of around 3291 per cm that actually represent the stretching vibration of the OH group. Okay, next we can see the CH aromatic, which is actually around 3075. for the CH aromatic. Then for the CH methyl group is around 1435. Then goes to the C double bond O group, which actually the stretching band appeared at 1667. Then we can observe the band at 1574, which actually represents aromatic ring group, and the stretching vibration for the CO group is actually around 1200. So the same goes to compound C2, but for the C2 is actually for the azocoumarine. Okay, so that's why we can see the presence of N double bond N, and also CN is to show the azocoumarine compound of the N double bond N and the C single bond N, which actually present at 1168.52 and also 1438 for this N double bond N. And for the CO group, is actually the band is actually expected at 108. And for CA is actually the metal complex. Okay, for the metal complex, this actually we expect this OH stretch is actually comes from the phenolic proton is loss of the formation of the formation of the complex metal. But what we can see here, the presence of 800 per cm is to show the coordination and the covalent bond ligand with central metal atom. So that is actually the the presence of the bond, the band at 800 per cm. Okay, and the rest will be the same thing for the CN and also for the NN to show the azo group or the azocoumarine, and the rest of the function groups are the same as your C1 and also C2. Okay. Okay. Next, okay, that will be your the FTIR characterization. Okay, next we'll go to the second spectroscopy method, which is proton NMR chemical shift data. For okay, now I just focus for this C2 ligand and the C8, which is the complex. So from the spectrum, okay, so this is the proton NMR spectrum. So we can see very clearly the sharp peak at around 13.71 ppm, which actually corresponds to the OH proton. And then next we'll go to this the proton signal at the range of 7.23 to 8.27 ppm is actually allocated to the aromatic ring proton. And also, apart from the aromatic ring proton, we also have the pyrone ring here proton. So the peak is actually observed at the area of 7.39 to 7.73 ppm. And then we have also the carbon or the hydrogen that attached to the carbon intercyclic compound, which actually represent the peak at 6.23, and then for the present of the 2.49 is allotted to the proton, methyl proton that attached to the pyrone ring. So this is actually the basic of the summary of these two compounds, the ligand and also the complex cobalt of C8. Okay, next. Okay, this is actually to have some more information of the peak of the C8 of the cobalt complex. So we can see very clearly the OH proton peak. We can steadily it become disappeared. So what we can actually have some conclusion here, we expect to have this OH proton peak for the proton aromatic, the proton cobalt complex, but it disappeared. So what we can have this conclusion is due to the deprotonation of the OH group. Okay, so the deprotonation, just to confirm that the OH is reacted to the cobalt 2 chloride that forming complexation. So then in the range of 7.16 and until 7.46 ppm, the peak is actually assigned to the CH aromatic ring, and 7.21 to 7.33 ppm correspond to CH pyrone ring. And then we have also for the cyclic carbon and also the methyl group that attach to the pyrone ring also show no difference either from the free ligand and also from the complex. Okay, all right. Okay, then we'll go to the compound of C3. Okay, C3. Okay, so this is the azo-coumarin compounds. Okay, the azo-coumarin. So again, same thing. We can see the peak of the OH proton. We also have this of aromatic CH for the pyrone ring as well, and also the methyl and the pyrone ring in an area of around 2.449. Okay, and for the C6, which actually the the copper complex. Okay, so again, the OH peak is also disappeared, as already mentioned. Before this is also due to the complexation formation of the copper towards the what we call this the ligand, the azo-coumarin ligand. So that's why we don't see any OH peak. Okay, and but the same thing goes to the aromatic CH, and also for the pyrone ring, we can still observe the peaks as what we can observe for the previous compounds of your C2, C8, and also C3. Okay, so this is actually the analysis of the proton NMR spectroscopy method to confirm that the product or the synthesized compounds already being synthesized. Okay, next, since we already been explained that all the 10 compounds being synthesized, so we try to have some propose of the mechanism to form the C1, or what we call this as a 6-hydroxy-4-methylcoumarin. Okay, so we can see that this is actually undergoes Pacman condensation method in order to form the C1 product. But the first step is actually the acid catalyst trans-esterification reaction, or we call it as a keto-enol tautomerization. So this is actually to show the first step where the starting material of the what we call this hydrocoumarin with the ethyl acetoacetate and undergoes the keto-enol tautomerization. And then we have, since the reaction is actually in the presence of strong acid, so we have this Michael addition. Okay, and the third step is where the dehydration process, where finally the first ligand of C1, 6-hydroxy-4-methylcoumarin, is already being produced and undergoes this dehydration process. So this is actually just some proposed of the mechanism of this product C1. Okay. So we finished the first part, which is actually the synthesis. So is there any questions on the synthesis part before we go to the second, to the third stage, because we already been, we have the method, first stage. Second stage is the characterization using 3 steps. Just now, as I mentioned, we have the CHN analysis, and then we have the FTIR, and finally the proton NMR spectroscopic method in order to confirm the presence of the product or the synthesized compounds. So shall I proceed to the third stage? After we finish all the products C1 until C10, we go to the anti-cancer screening activity. Okay, but okay, we have all these 10 compounds just now, as I mentioned, C1 until C10, but we try to minimize the cost. Since anti-cancer screening activity, we have to send to this Institute Biology at University Putra Malaysia. So we just choose out of 10, only 4 compounds, okay, which is C2, C2 is actually the ligand of isocoumarine. C8 is actually the what we call the complex, the metal complex. C3 is also the metal, the isocoumarine, and C6 is the metal complex. But what's the difference between C3 and the C8 is the type of metal, which is copper and also cobalt. And then why we choose C8 and also C6, because we try to compare the same halogen, because just now we can see the halogen we chose, we have 3 types of halogen, which is the chloro, bromo, and nitro, and we conclude to have this, just focus on the halogen group of the chloro. Okay, so just focus of this isocoumarine where the presence of the halogen is chloro, meaning C2, C8, C3, and C6. All these compounds is actually consist of chloro, but C8 is actually the what we call it, the metal of copper, and C6 is actually the metal of cobalt. Okay, the other way around. C8 is the cobalt, C6 is the copper. Okay, now, okay, so these 4 compounds, okay, we have this, we undergo this anti-cancer screening. So we send it to the, what we call this Institute of Biology in University Putra Malaysia, because in USIM you don't have this kind of instruments, okay, to test the anti-cancer screening. Okay, so I try to have, to inform that the cancer cell that is being used, we call this MCF7 cell, is actually human breast adenocarcinoma cell line estrogen receptor. Okay, so this is the type of the cell. Okay, basically it's a human breast cell. And from the complete results, okay, from all the 4 products or the 4 synthesized compounds, the ligands, the C2 and C3 is the ligand, okay, the isocoumarine. Okay, and the complexes of C8 and C6 inhibit the cell proliferation. And what we can conclude here that MCF7 cells were treated with different concentrations of the compound, okay? And the MTT assay that is used is to determine the percentage cell viability. Okay, so the question here, what is actually the percentage cell viability? We use this formula where we have the absorbance sample, get the mean, and divide it with the AC, which is actually the absorbance of the control. And then we times this 100%, and that will get how the percentage cell viability. So what is actually the percentage cell viability is actually we can define as the ability to of the cell to survive. So if the percentage of cell viability is low, so we can say that the survived cancer cell is low. And thus it shows that the higher the anti-cancer activity of the synthesized compounds. So this is actually how it's being interpreted by the percentage of cell viability. And then next, okay, so what we did, from the MTT assay using the ELISA reader, so we get this optical density or the OD value. And from the OD value, we get the absorbance, okay, so this is the absorbance, okay, together with the standard deviation. Okay, and these are the percentage from the absorbance. We can get the percentage of viability for all the 4 products or the 4 compounds, C2, C8, and this is the C3 and also the C6. Okay, from the absorbance of the optical density, using the formula that I mentioned before, to get the percentage of the viability. Okay, all right, so what we can have some summarize from this percentage of viability. So we can see from the data here, there's a wide range of absorbance of the optical density of the compounds. It's actually been measured using ELISA reader. And the widest range of the absorbance was at the wavelength of 570 nanometer. So we just focused on this wavelength, okay, 570 nanometer, and we can see the range of the absorbance is from 0.145 until 1.767. And for the control, okay, so we can see we have the control here. So the absorbance values that are lower than the control cells indicate a reduction in the rate of the cell proliferation. As contrast, a higher absorbance rate indicates an increase in the cell proliferation, and all the absorbance values are lower than the control cell, which actually indicate the reduction of the proliferation. Okay, so basically this is actually the summary of the OD, okay, and the percentage of cell viability of the 4 compounds. Okay, then I extract this value to this. Okay, so from the 4 compounds, or the C2, C8, C3, C6. So these are the percentage cell viability. So what we can see here, okay, so we can say the percentage of the cell viability for C2 is 22.12, C8 is 19.33, C3 is 8.20, C6 is 10.71. So we can see at this concentration, okay, we actually comparing the two here, C2, the ligand, and the complex, C8, and then to the same thing, the C3 is the ligand, and the C6. So what we can see here, at this concentration, the viability of the cancer cell of C3, okay, which is 8.20, is lower than its complex of C6. However, for compound C2 and C8, the viability of cancer cell in the cobalt complex C8, it seems that the value is lower compared to the ligand of the C2. All right. Okay, so then after we get the, what we call this now, the percentage of cell viability, then we'll go to this value. We call this as IC50 value. What is actually IC50 value is actually the anti-cancer activity that is recorded as drug concentration that cause 50% growth inhibition of cancer cell. So after we get the, what we call this now, the percentage of cell viability, then we go to IC50 value, meaning that we don't use the just one value of the percentage of cell viability, because what is the most important part is the IC50 value, because this is the main what we call this parameter to show the anti-cancer activity. Okay, so next we'll go to this IC50 value, and we summarize all the IC50 value and how the IC50 value is actually being introduced is having this, okay, let's say we have this graph, okay, this is actually one of the graphs to show how the IC50 value is being determined. So let's say we have the C2 compound, okay, how the 43 microgram per milliliter is being observed is where we plot a graph of the viability, the percentage of viability from 0 until 100% to the, and the concentration, okay, and we get the 50% because just now the percent, because what it means by IC50 value is the 50% growth inhibition. So from the 100% viability, we get the 50%, and then we will get the value of the concentration. Okay, so for C2 is 43 microgram per milliliter, for C8 is 29 microgram per milliliter, and then for the ligand of C3 is 32, and for the C6 is 35. Okay, so what we can discuss from this value of C2, C8, C3, and C6. Okay, now, so from here it can be determined that the IC50 value of C2, okay, as 43, after, okay, before I proceed, this is actually the exposure is actually covers until 72 hours, okay? So after 72 hours exposure, so we can determine the IC50 value of the C2 of the C8, which is actually 29 microgram. And from the comparison, we can see that the IC50 value is decreased from 43 becomes 29 after the complexation of the C8. Okay, therefore, what we can conclude here, okay, from the IC50 value, since the C8 is smaller, which is 29, compared to C2, so we can conclude that C8 is more potent anti-cancer agent compared to its ligand, okay, which is only is 43 microgram per milliliter. Okay, then we'll go to the compounds of C3 and C6. Okay, for C3, okay, it seems that the IC50 value is 32 microgram per milliliter. However, for the complex, copper complex of C6, it shows that the IC50 value is 35 microgram per milliliter after 72 hours exposure. So what we can see here, that the IC50 value of the ligand, okay, is lower compared to the copper complex. Okay, so what is actually being discussed here? Okay, so from the compound C2, C8, C3, C6, we can see the presence of hydroxy, or the hydroxyl group, which is actually at the position of 6. So it seems that the presence of this hydroxyl group also can affect the activity of the ligand by decreasing the IC50 value, okay, and due to the electron affinity and high electronegativity. And therefore, it can be assumed that will be the reason why the lower of C3 ligand compared to the copper complex of C6. So we have these two about what we call the contradict between the copper and cobalt together with the ligand itself. Okay, all right. So, so what we can see here, what is actually IC50 just now is actually the 50% of the cell inhibit, but we can also add some information about IC50 value. Is actually the pharmacokinetic measure of a potency of a substance and inhibit a specific biological function. So in this case, the cancer, the cell cancer, which is the MCF7, and IC50 also can be defined as the drug concentration, whereby 50% of the target is inhibited. And from here, we can also conclude that IC50, the lower the IC50 value, the stronger the inhibitor and more potent the drug. In this case, the drug is actually the 4 synthesized product. Okay, so now, so this is actually the summary just now. I just show back again. From here, the IC50 value of the out of the 4 synthesized compounds, C8 will be the lowest, which is only 29 microgram per milliliter. So we try to have some conclusion that C8, which is a cobalt complex, gives the most potent anti-cancer agent. Okay, why? So this is actually some of the, what we call it, recommendation or some proposal why it gives the most potent anti-cancer agent. Because from the studies from Tamila Rasain is actually one of the researcher also focus on this anti-cancer. Okay, it shows if the certain compound, if a certain product gives most potent anti-cancer agent, it shows that that particular compound has the highest ability to bind and inhibit the DNA replication, which is actually focusing on the cell, MCF7. So then how it inhibit to bind and to bind and inhibit the DNA replication? By killing the active growing cells through the release of chloride ion inside the cell. And then the species of the ROS, or the reactive oxygen species, being generated, which can induce the cell death. And the compound also we can see from the C8 is a para substitute also exhibit a higher anti-cancer activity because it has high electron affinity because of the presence of the chloro just now, and high electronegativity. All this, the presence of chloro will lead to high reactivity of the compounds in inhibit the species. And then not only the halogen or the what we call this, the para-substituted halogen group, not only the hydroxyl group, we also can see the benzene ring. Okay, the benzene ring of the para-substituted halogen also more active because of the halogen group at this position will actually give more active to the ring, and thus increase the inhibition process of the cell proliferation. But from the studies here, we just try on the presence of the halogen of chloro. We haven't finished yet on the studies of the bromo and the nitro. So I think after the next study, we can compare between the halogen group of chloro, bromo, and nitro, which one will give the best to exhibit higher activity, anti-cancer activity. From here, we can only conclude, because we are comparing the same halogen group, which is chloro. But next, we try to have to compare between bromo and also nitro, what happened, the effect of the halogen. Okay, and also we try to have some, what we call this, the metal complex, not only cobalt, also copper. We also try to have some other metal complexes. For example, we have also nickel, and we try to have some comparison between all these metal complexes. Okay, so as a conclusion, okay, we have already successfully synthesized 10 compounds of 6-hydroxy 4-methylcoumarin derivative and also the complex. And all the synthesized compounds have been characterized using CHN elemental analysis, FTIR, and also proton NMR spectroscopic methods. And from the conclusion and from the characterization of the FTIR, it shows the important functional groups presence in the compounds of the OH, of the C double bond O, of the C double bond C, and for the C and for the azo group, azocoumarin. Then when we look at the characterization of the proton NMR analysis, it shows or it possesses the presence of proton signal, okay, in the presence of 7.23 that allocated the proton aromatic, the proton methyl, and the proton pyrrole respectively. And so we have the characterization. We go to the anti-cancer screening activity of the 4 compounds, C2, C3, C8, and C6. And it revealed that the reduction in the rate of the cell compared to the untreated control cells, it gives, or it shows all the compounds shows the potential as anti-cancer agents. But between all the 4, we can see only C8 will give the most potent anti-cancer agent. And what we can conclude here is due to the presence of the chloro substituents just now. Okay, chloro substituents and the complexation of the metal of cobalt, which actually gives a very important, or it gives play important roles in the anti-cancer activity. Apart from that, also, as I mentioned before, all the 4 consists of the substituent of halogen group, which actually affect the anti-cancer of the synthesized compounds, as it has high electronegativity, electron affinity, and high electronegativity. Okay, so these are the references from our studies and also the acknowledgement of the financial support from the USIM grant. And if there's any question, I'll be here to entertain the answers. So thank you very much for the presentation.
Okay, thank you, Juliana. It's kind of inspiring presentation. From Dr. Juliana, we know that organic compounds have biological activity, anti-cancer, especially for breast cancer, that maybe most of the patient is women. Okay, once again, thank you for the presentation. We are now going to begin answering the questions submitted during the presentation. And we already have several questions. May I share the screen? The first question is from Sulaksana Permana, Center of Mineral Processing and Corrosion Research, Department of Metallurgical and Material Engineering. And the question is, as you said in the previous explanation that coumarin derivatives has its anti-cancer potential in human body, my question is, how does the coumarin derivatives work as an anti-cancer, and may the honorable speakers explain the mechanism for coumarin to be an anti-cancer in human body? Thank you very much. Okay, Professor.
Okay, thank you for the question. Actually, okay, so this is actually the preliminary study, okay, the preliminary study when we try to have the to synthesize a compound, and then after we synthesize a compound, we'll go to the anti-cancer screening. Okay, meaning that the anti-cancer screening is just to screen, meaning that from the 4 compounds, okay, we have the C1, sorry, C2, C6, C3, and C8. Okay, we just screen, and from the screening anti-cancer, we get these 2 values just now. We have this percentage cell viability and also the IC50 value. Okay, remember just now, and from the final results, it shows that the IC50 value from the results, the lower the IC50 value, the higher the potent as the anti-cancer activity. But regarding the question just now, the mechanism for the coumarin to be an anti-cancer in human body, we have never started the study on the mechanism of the coumarin. We just started how the synthesized product, the mechanism of the synthesized product. After this, then we try to get the mechanism how the anti-cancer it affects the anti-cancer activity on the human body. But from the comparing, because that's why we select out of 10 just now, we have 4 just now. So from these 4 compounds, okay, we can see that, okay, we have the complex. Okay, the metal complex itself. So what we can conclude, we just have some first, what we call this conclusion. The presence of the metal can give more potent towards the anti-cancer activity. But how the mechanism is actually the further step. Because after this, we try to have more explanation for the postgraduate student on how, but because this is actually the undergraduate, we just have some information regarding the structure of the product, meaning that just have some substituent, okay, the presence of hydroxyl group, the presence of the halogen group, the presence of the metal. But now we just prove because of the presence of the metal, okay, it gives towards the cancer activity. The mechanism started yet. So this is actually the further studies where we try to have some studies on the mechanism. Okay, why or how the anti-cancer and the compounds can affect the studies on the anti-cancer. Okay, so meaning that for this question, we can have some more studies on how the mechanism for the coumarin can affect the what we call this anti-cancer activity. But what we can see here, just the presence of the halogen group, just the presence of the metal group. Okay, but we try to have some further studies, the mechanism of how the structure, but what, as I explained just now, we just have some proposal just now, as mentioned in the final slide just now, and only inhibit, okay, inhibit the what we call just now the DNA cell just now. But how the studies, this is actually a further studies that actually should be conducted for the postgraduate students. Okay, sorry for not having some explanation on the mechanism on how it affects the anti-cancer. Is that okay, Sulaksana Permana?
Mr. Sulaksana Permana, is that okay? Okay, we move to the second question.
Okay, thank you.
Okay, thank you. From Putut Universitas Islam Indonesia, I would like to ask if the synthesized compound will be able to become a potent chemotherapy agent. Can this compound be classified as a cytotoxic agent, and how is the mechanism of this compound so it can specially affect cancer cell without damaging the surrounding normal tissue? Do you transfer it?
Okay, this is also actually the question. The second question is also almost the same, because how the mechanism can affect the cancer cell, because I haven't gone through the studies on this cancer cell yet. But for the first part, the first question, is the synthesized compound will be able to become a potent chemotherapy agent? Okay, so because actually the next plan, apart from this anti-cancer, we also in University Putra Malaysia also have this test, or we have the screening on a chemotherapy agent, and also we have this cytotoxic agent. Okay, so for the cytotoxic agent, I would say that, well, because cytotoxic agent or and anti-cancer agent is almost the same, we can say that if the cytotoxic, if the anti-cancer is good, gives a positive anti-cancer good, so it might be the cytotoxic also gives a positive cytotoxic agent. But for the chemotherapy agent, we haven't studied yet. But I think this is one of the, what we call this, idea, or some what we call that advice, or maybe idea to have more further studies on this chemotherapy agent. So might be, but because we haven't studied that. But for the cytotoxic agent, yes, I have already explained before. If normally, if the cytotoxic is good, meaning that if IC50 value gives a low IC50 value, it can also conclude that it also gives a cytotoxicity activity. And thus, we can see here is also a potent anti-cancer cell activity. Okay, so for the chemotherapy agent, I think we can have some more studies on this chemotherapy, because we haven't started yet. For the time being, we just started on the anti-cancer agent, the potential. But how the potential of anti-cancer is from the cytotoxic activity. So that's why we can say that well, can be classified as cytotoxic activity, because from the studies it shows that how the cytotoxicity activity can be determined. It's also from the IC50 value. Okay, so meaning that it will be the same method on how to determine a certain compound can be also considered as a potential as activity, can also from the cytotoxicity activity, because the method is the same, meaning that just use from the MTD assay, you get the percentage of value with IC50. So just can confirm the second one, the cytotoxicity activity. And for the last one, the mechanism, as already mentioned before, using the same question, I think how it can specifically affect the cancer cell. This is a very high or very more further studies where I will have some recommendation for my postgraduate student to study the mechanism itself, how the compound of coumarin can affect the cancer cell without damaging the surrounding normal tissue, because this is a very detailed studies. But for this study, since this is one of my undergraduate students, they just study on the screening, on what we call this anti-cancer screening activity. So I think next time, if I was invited by Prof. Fatima, maybe I can get more information or studies on the mechanism of this compound and how it can affect the cancer cells without damaging the surrounding normal tissue. Because this is a very detailed studies, because it also involves biology unit. Okay, because our studies, my students, undergraduate, just focus on the what we call this organic synthesis, just the synthesis. So next, I think we will try to have more information, more studies on this. Okay, I think that would be my best explanation for Putut. Is that okay, Putut? Or maybe we can have, okay.
Okay, got the point. Thank you very much, Dr. Juliana.
Yeah. Maybe we can have some, if let's say you also involved in this, maybe we can have some, what we call this, joint venture to have the studies on the basic specific on the effect of cancer cells without damaging the normal tissue cell.
Okay, that's a great idea. Thank you.
Okay, thank you, Dr. Juliana. Maybe Dr. Juliana can have collaboration with Dr. Tatang Sapurung Sulianto. So it can explain the mechanism by molecular docking.
So Dr. Tatang, maybe you can have some explanation or ideas, because my students is just focus on the preliminary studies, just have some, what we call this, the screening anti-cancer. So just focus on how the effect of the structure of the compound, but we don't even go further to the cell or the mechanism itself. Maybe Dr. Tatang have some ideas?
Maybe later. Not now.
Or some, maybe we have some collaboration after this to get this mechanism, because just for this first studies on this, just have some screening. But I think we should have some more explanation on how the detail of the mechanism that affect the cancer cell, right?
Be patient.
Okay, Dr. Juliana, this is the last question from Sarah Ayu Wulandari. I would like to ask, what is the effective level needed to consume the coumarin as an anti-cancer agent? Because according to the source that I read before, coumarin compound is banned in US since 1954 because it can cause liver problems in high concentration. Thank you for the opportunity.
That's why, if you can see just now, okay, if you see the studies just now, the concentration is very low. Okay, it's very low because of course we have some limit on the concentration. So if let's say we have the effective level, so if you can see the studies just now, if you can I have some, how can I share the slide just now? My slide. Miss Lena.
Yes.
How can I share my slide just now? Okay, can I share my slide just now? Okay, all right, okay. So to answer this question, if let's say, okay, okay. So since we did, okay, if you can see for this, oops, enter cancer. Okay, so, okay, okay, so these are the concentrations. Okay, so you can see it's microgram per milliliter. It's a microgram per milliliter. That's why it's very low. So hopefully after we have a further studies on the concentration, we might be able to get some information, the minimum, because our area is just to get some values of concentration. Very low is microgram per milliliter. But in order for you to have some level here, I think we have some get some further information regarding the what we call the level of the concentration of the coumarin. But what we can see here, the concentration that we did, okay, is from 1.56 microgram per milliliter until 100 microgram per milliliter. Okay, so I'm not sure regarding the level of the level that is actually being required, okay, from the government. But this is also something that we have to consider. We have to study back regarding how it can actually affect to the bad health just now, as mentioned by one of the participants just now. Okay, okay, I think that's all. Any other questions?
Okay, thank you, Dr. Juliana. May I share the screen?
Okay, thank you.
This is a question from Arif Ahmadi, Universitas Tadulako. I think this is the same question, and the answer is Dr. Juliana.
Oh, the question regarding how the mechanism, is it?
Yes.
The mechanism that actually affect. Okay, this is might be also with get some, you know, studies on the mechanism actually can you hear me?
Yes, Dr. Juliana.
Okay, can you hear me? Okay.
Yes.
So the answer will be the same thing?
Yes. Thank you very much for the nice
Okay.
presentation. And
okay.
now we can move to the second speaker. Dr. Tatang Sabur Julianto from Chemistry Department of Universitas Islam Indonesia. Let me introduce him for a moment. His research interest is drug discovery from natural product, microbial and enzymatic food modification, pesticide development from natural product, biofuel development from biomass. And besides this project activities, he remains to take his time to write many books. You can look at the screen. There are 4 books, and maybe we can see that he wrote about essential oil, biochemistry, anti-malarial, and phytochemistry. Okay, now without further ado, we will turn the time over to Dr. Tatang Sabur Julianto. You have 30 minutes and the screen is yours.
Okay, thank you, Ibu Lina. The Honorable Professor Isfatima and also Prof. Rui that inviting me in this webinar. Also to my colleagues, Mrs. Juliana, Dr. Juliana. It is such a coincidence. We have the same phrase of name, yeah, Juliana and Julianto. It means we born in July. Okay, but I'm wondering why your profile name is Kamal. So,
because I'm using my husband's laptop.
Okay, thank you. Okay, thank you to Ibu Lina that have introduced myself. My name is Tatang Sabur Julianto. I was born in Pamekasan, that lie in small island in East Java, Indonesia. Yeah, and then my education, I was graduate from chemistry of department Diponegoro University in Semarang. My research is about the natural product of isoflavone also in anti-cancer agent, just like Mrs. Juliana. And then I finished my master and doctor in Gajah Mada University in organic chemistry, especially in anti-malarial drugs synthesis, from phenanthrolene derivative and also in aryl amino alcohol, and try to synthesize something, some pathway of artemisinin also. And then my research interest, just Mrs. Lina said that I interested in drug discovery from natural product, also in microbial and enzymatic food modification and pesticide. And the last is the biofuel development from biomass. Okay, this is that I identified from some journal of organic chemistry, that the current scope of journal that say that tell about the new synthetic method and methodologies, isolation and synthesis of natural product, also in organic reaction, bioorganic and medicinal chemistry, and so on. But today I would like to share another perspective of organic chemistry that that's just Mrs. Lina said that organic chemistry is scary, scary field of chemistry, just like the meme that you have saw before. Okay, this is the good challenge of organic chemistry, that we need to would reside at the interface with the energy, environment, sustainability, material, biology, medicine, and also in computer science, to help us predict the activity, just like molecular docking analysis. Okay, allow me to deliver the presentation in bahasa, also in English, campur-campur, mix, so we can get good interaction with the audience also. And this is my presentation today about the role of bioconversion in isolation and synthesis of natural product. Okay, Indonesia is the one of the country that have the largest rainforest, the third world largest forest, so many biodiversity there. So Indonesia rainforests are home to some of the highest level of biological diversity in the world. Many sources credit Indonesia as the most species-rich country on Earth. And we have 17,000 islands that contain of the area of the rainforest. So this is some of biodiversity species in Indonesia, including animals and flora. And then, okay, we start from the natural product definition, because a natural product is also the branch of the organic chemistry. When we have the definition, natural product is the chemical compound or substance produced by living organism. Then the natural product can also be prepared by chemical synthesis, both semi-synthesis or total synthesis, and have played a central role in the development of the field of organic chemistry by providing challenging synthesis target, all starting material also. And then the term natural product has also been extended for commercial purposes. Just like in cosmetic, we know that cosmetic with the source of the natural product, and then dietary supplement and food that produced from the natural sources without added and artificial ingredient. Okay, this is the secondary metabolite. The content of the natural product within the field of the organic chemistry. Then the definition natural product is usually restricted to the organic compound. The organic compound isolated from the natural source that are produced by the pathway of the primary and secondary metabolism. Yeah, within the field of medicinal chemistry, the definition changed into the secondary metabolite only. Yeah, and then the secondary metabolite are not essential for the survival, different with the primary metabolite. Yeah, but I think this is very important to evolutionary advantage for the organism. Yeah, that metabolite, the secondary metabolite has been produced by organism to have evolutionary advantage. Yeah, just like here for the inhibit the parasite and predator or herbivore. Yeah, before and then be the pollinators. Yeah, and then the others. Yeah, or protect from the thermotolerance, yeah, with the sun. Yeah, the sun. Okay, many secondary metabolite cytotoxic and have been selected and optimized through the evolution for use chemical warfare again prey, predator, and combating organism. But when we have the the right doses, yeah, it will be not toxic anymore, but we can be drug for the human or for other human living organism. Yeah. Okay. Next, this is the primary metabolite. I will be not explain this one because I will be focused on the metabolite secondary only. So yeah. And then this is the example of the some secondary metabolite that already used by the human the organism, yeah, by organism. And then the global trend right now, the year in headlines, yeah, moved into the natural and organic. Just like, for example, Unilever that to acquire Brazilian heritage organic brand. Yeah. Also, the Campbell's acquired the organic food pioneer. Yeah. So the organic or natural product will be important in the next future. Yeah, in our life in the next future. Okay, first, I will try to tell you about the natural product in drug development. Yeah, this is how the natural product has been isolated to be the lead compound. Yeah, and then with the synthesize, we can produce the natural product analog, just like in the originated by the natural product. Yeah, that have already test the bioactivity to be a drug. Yeah, to be a drug. Not only from the from the plant, also from the just like frog or fungi, yeah, or other living organism. Yeah. Okay. So this is one of case that one of the famous drug that we call by Tamiflu, yeah, originated from shikimic acid. That shikimic acid can be extract from the Chinese star anise. Yeah. So not all of the drug can be synthesized from the the starting material that not from the natural product, yeah, but also from the natural product. Yeah. Okay, this is the other source of the drug that called by salicin, originate from the willow tree. Yeah, willow tree. Salicin can be converted into the aspirin drug that have the analgesic agent. Yeah. Also in natural product in food, not only drug, a natural product can be developed to be the food ingredient, just like a flavor or flavor, and then this is a kind of the flavor, natural vanillin, natural vanillin can be isolated from the vanilla pod, but today vanillin can be produced by bioconversion or total synthesis, from other sources, just like rice bran, and then the eugenol, also from the turmeric, curcumin, and also the glucose, can be converted into the natural vanillin also, not natural, but synthetic vanillin, I think. Also, natural color for food, just like carotenoid, flavonoid, beta-alanine, also in chlorophyll, we have the anthocyanin also here, the different structure give the different color, also effect by not only the structure, also with the pH value, that the different color of the anthocyanin will be affected by the different pH also, just so in this slide, can be red color, can be, what is it, purple, also in blue color, also in the yellow color, Okay, this is, so the anthocyanin can be a potential to be the indicator of the pH. Okay, another role of the natural product also in agriculture, This is the product of the natural product to be the pesticide, from neem tree. In Indonesia we call by mimba. Maybe some person already know about this plant, when I go to the Jabal Rohmah, also I meet this tree also, they called by Sukarno tree, because President of Sukarno have bring this tree to Saudi Arabia, and in our town, the neem already convert to be jamu, just medicine, traditional medicine in Madura, jamu Madura, Okay, but in the for agriculture, azadirachtin can be act as a pesticide, pesticide is commonly in India, I think, also to be health and medicine also, and cosmetic, This is the natural product that I found potential to be the pesticide also, from the leaf of Mahkota Dewa, Valeria macrocarpa, that I found and isolated the alkaloid compound that called by narcotin, from the leaf, that I try to applicate to the Spedoptora litura, one of the insect, the insect, that it will and have a nice activity, Also, natural product can be natural repellent, just like for the repellent of the mosquito, from the peppermint, lavender, basil, rosemary, citronella, and marigold, But in the development of natural product, we have several problems. First is incompatibility of crude extract with the test procedure, So, and then the other is a lack of the reproducibility result, high cost of isolating natural material samples, and then difficulty of in isolating active compound from extract, problem with the large scale supply if drug arise from the natural sources. Also, slow growth, slow growth, species and distribution and spare species and transfer the Yes, this is the sum of several problems ya, in natural product. Jadi cukup mahal ya. Jadi mengisolasi senyawa dari dari bahan alam atau semula jadi gitu ya, itu sangat mahal ya. So we can choose total synthesis to create the same compound of a natural product ya. As we shown in my slide, this is the structure of artemisinin. Artemisinin is the main anti-malarial drug right now, recently. So yeah, recently this is the crystal of the artemisinin that can be isolated from the Artemisia annua. Artemisia annua only growth in subtropical countries, just like in China and Vietnam, the North Vietnam, also in other country that have the subtropical country. Yeah. Otherwise, there are no malaria case. Yeah. So when we try to grow this Artemisia annua in Indonesia, not good obtain the artemisinin. So we still need to import from other country. So this is the some researchers already try to give a total synthesis of the artemisinin from isopolygol, just like Smith and Hausen has been done. Yeah. Also Yadav in 2010. Yeah. A long pathway. Yeah. 11 step, 8 step, 5 step, and this is the 21 step. Yeah. So that make the synthesis to be expensive development. Yeah. So why the Chinese still try to isolate from the Artemisia annua. Okay, this is my proposal to synthesize the artemisinin also with the short with the short step. Yeah, only 6 step. Yeah, with the what is it? With the easy starting material. Yeah. Yeah. Okay. Okay. Synthesis of the natural product can also used to modify the chemical structure in enhancing the bioactivity, just like in artemisinin also. Yeah, I compared only the artemisinin. Artemisinin have a high toxicity, so with the modified chemical structure to be artemeter, yeah, just like this structure. Yeah, so the toxicity is decreasing. Yeah, decrease. Okay, this is the other the other aspect of the natural product about the development of isolation technique. Yeah, we try to toward the green extraction. Yeah, green extraction. This is the purpose or goal to have a green extraction technology. Yeah. This is what I have done. Try to do isomerization reaction without solvent. Yeah, solvent-free isomerization. Yeah, and then so this is what I want to explain today. Yeah, about the role of biotechnology in natural product development. Yeah, first, biotechnology can be act in isolation and bioconversion. We call by green extraction here, green extraction, green. Also, we use genetic engineering, but this is not our field, yeah, because it is for biologists, I think, yeah. Okay, first. About the bioconversion in isolation of natural product. So I divide the bioconversion into two group. Yeah. First is for the isolation or extraction, yeah, of the natural product, and second is bioconversion for the synthesize, yeah, for the synthesize. Okay, first for the bioconversion in isolation of the natural product. Usually, bioactive compound are recovered from natural source by solid liquid extraction. Yeah, just like chocolate extraction or others. Yeah, employing organic solvent in heat reflux system. Yeah. So, however, other techniques have been recently proposed to obtain this compound, including the use of the supercritical fluid, yeah, just like I explained before, yeah. Also in high pressure processes, microwave assisted extraction and ultrasound assisted extraction, yeah. And then extraction or production of bioactive compound by fermentation is the alternative. Is also an interesting alternative that merits attention, yeah, since it is able to provide high quality and high activity extract. Yeah, we found racemic in what is it? The conventional extraction method, yeah. But when we use fermentation or bioconversion, yeah, it is a very interesting alternative, yeah. That in this process, bioactive compound are obtained as secondary metabolite that produced by microorganism after the microbial growth. Yeah, it's complete. Okay, this is a case of the use of bioconversion in isolation. Yeah, when this is what is it? The eucalyptus leaf. Yeah, when we do the hydrodistillation of the essential oil before we do the distillation, sometimes we need to do the preparation before, yeah, distillation, yeah, with fermentation, yeah. The fermentation make the yield of the oil will be increased, yeah. This is in 2 days, 4 days, and 6 days, yeah. Okay. Okay. And then this is this is one of my research, yeah, the isolation of the ferulic acid from the brain, sorry, not brain, yeah, brain is my brain, yeah, brain, yeah. Okay. Okay, this is why I choose fermentation, yeah, because we need to hydrolyze, yeah, with the green extraction, yeah, using what is it, the bacteria. The bacteria, in my case, I use Lactobacillus plantarum, yeah, Lactobacillus plantarum, that we can release the ferulic acid from the structure of the brain, yeah, from the brain. That the brain is containing the hemicellulose, lignin, also the structural protein, pectin, and the phenolic acid, just like ferulic acid. Yeah, ferulic acid is what is it? The compound that can be changed into another compound that have available economically. Yeah. Okay. The other is use enzymatic assisted extraction. It will be specifically, yeah, the structure of the cell membrane and cell wall, micelle formed by macromolecules such as polysaccharide and protein, and the coagulation and the denaturation of the protein at the high temperature during the extraction are the main barrier to the extraction of natural product. So we can change into the EAE, yeah, assistant extraction. The extraction efficiency will be enhanced by due to the hydrolytic action of the enzyme on the component of the cell wall and membrane. Yeah, we know the cell wall construct by the cellulose, yeah, and the membrane by the lipid, yeah, by the lipid, and the macromolecule inside the cell which facilitate the release the natural product. For example, we can use cellulose, also alpha amylase and pectinase in are generally employed in EAE, yeah, enzymatic assisted extraction. Yeah. Okay, this is one of the pathway how to hydrolyze the material of the natural product. Yeah. And the second, bioconversion also can use in apply in a synthetic, in synthetic of the natural product, synthesis of the natural product. So we need microbial bioconversion is the process by the microbial spores or cell used to convert the organic compound to a structurally related compound through one of the few enzymatic reaction. Okay, well, one of the earliest example in our life is about vinegar. Vinegar can produce from ethanol, ethanol that it produced by acetic acid bacteria, just like acetobacter and gluconobacter, gluconobacter. This is the one of the natural products synthesized by bioconversion, the first, Also, acetic bacteria involve the bioconversion, not only ethanol, also the other alcohol compound, just like propanol, isopropanol, convert to be the what is it? The acetic acid, propionic acid, acetone also, oxidation, the oxidation reaction. Okay. So some of the advantage of the chemical synthesis can be can be act by the bioconversion method. Several disadvantages chemical synthesis follows. First, low catalytic efficiency. Sometimes we have a racemic mixture in the product of the synthesized, so we need purification process. Also sometimes lack of enantiomeric specificity or for chiral synthesis, and then need for high temperature, also low pH and high pressure. So when we move into the bioconversion, there are some advantages of bioconversion. First is the biocatalysis has a higher enantiomer selectivity and higher radioselectivity in aqueous solution, and then does not acquire protection and the protection of functional group just like in organic synthesis. So that has better stability also, and operate under mild condition. We do need high temperature, or lowest temperature, low temperature. That is avoidance the high and low temperature, and then has a greater efficiency, because we use enzymatic activity here, and higher product yield can be produced, is about 90 to 100% based on the Tau and Zoo experiment. Okay, for example, this is the terpenoid structure, limonene. Limonene can be changed into another compound, that have a commercial value, only by bioconversion. For example, limonene can be converted into the carpeol, by Fusarium proliferatum, also can be changed into another, just like terpeniol, this is another flavor or fragrance, or fragrant compound, just like terpeniol, just with the applicate the aspergillus niger in limonene. Okay, it's about the yield is about 47% also. Okay, this is another example about the terpene bioconversion. We use microbial degradation of the citronellol and nerol by Pseudomonas citronolysis, to be another compound. Also citral and citronella here by Pseudomonas aeruginosa. This is some bioconversion reaction using flavonoid as a starting material. First, we can do the hydroxylation by bioconversion. Bioconversion, just like in this slide. Hydroxylation. This is the kind of the microorganism that can use for the hydroxylation reaction. For flavonoid. Already obtained by some researchers. And then also you can do the methylation. Methylation in conventional synthesis we use just like hydrogen bromide or BBr3 in DMC, also in lithium chloride, in DMF by reflux, methylation. But when we use bioconversion, we have a mild condition. We don't need organic solvent or also some reagent that I think is very expensive to have. Also, or the methylation, with aspergillus niger, This is the kind of the microorganism. Another except the aspergillus niger, this is what I've done. The hydrolysis anthocyanin to be anthocyanidin by Rhizopus oligosporus. This is the fungi, the fungi microorganism originate from the tempe, from the tempe ragi, from the tempe yeast, from the tempe yeast. Okay. So I have done 5 days of fermentation time. This is the extract, and then this is the reaction of the enzymatically beta glucosidase, that already arise the pelargonidin containing inside the extract. So the glycosidation, deglycosidation of the flavonoid glycoside. This is the flavonoid glycoside that we call by anthocyanin, to be anthocyanidin, improve the antioxidant activity 10 times until 20 times more active of the antioxidant. Also, I have done the production of lactone by bioconversion. We know the lactone flavor with the fruity, milky coconut flavor, or aroma, widely used in the food of fragrance industry. Lactone is the one of them. Lactone are produced by chemical synthesis usually, but we can change into the biotransformation or bioconversion, using the Saccharomyces, something just like I have done. We have the delta octalactone here. This is the mechanism how can lactone produce. First, the starting material is fatty acid. Fatty acid originated from the fat. Fat can be hydrolyzed to be fatty acid by bioconversion or by the acid. The acid, and then we do the hydroxylation with the bioconversion. With the Saccharomyces also. And when the fatty acid synthesized can be convert into the acetic acid, or acetyl CoA. We call it acetyl CoA when the beta oxidation happen in the metabolism of the fatty acid. And then when the beta oxidation happening, then we move into the lactonization. So we have a dodecalactone here, gamma dodecalactone. Okay, for the conclusion, I think modern research has focused on the microbial transformation right now recently, of the huge variety of the organic compound to obtain compound of therapeutic and industrial interest. Also, bioconversion or microbial transformation is useful tool, for the producing new compound as a consequence of the variety of the reaction of the natural product. Okay, that's all. Thank you for the time. I over to the moderator. Miss Lina, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Tatang. Okay, once again, thank you for the presentation. We are now going to begin answering the question. Let me share screen. The first question, Dr. Tatang, is from Dr. Zulkarnain Jalil, Universitas Syah Kuala Banda Aceh. Aceh known as the biggest and best quality for ganja. In the point of scientific view, it is a possibility to extract a new type of drug from our ganja. Thank you. It's nice question, interesting.
Yes. Yeah, okay. Thank you, Dr. Zulkarnain. I'm sure you have already extract the ganja, yeah. Or, I don't know, because Banda Aceh is the central of the ganja, the pusatnya ganja ini ya. Kalau ganja terbakar ya, nanti kalau anginnya ke mana? Ke Singapura, orang Singapura bisa mabuk semua itu Pak ya. Okay, I think it's possible to extract the ganja Yeah. I don't know what exactly the metabolite secondary metabolite in ganja yeah, but I think it's about alkaloid alkaloid compound I think yeah. Maybe with the modification of the structure of the alkaloid in ganja, we can change the what is it? The toxicity or a negative effect of the ganja to be another bioactivity that can use to be drug, to be drug. I think yes, it is possible. Thank you. Another question?
Okay, the next question from Ida Dumariris. One of the most popular today is herbal plants that can increase antibodies. What parameter can we use for this? Maybe the mean is what parameter that we can use to know which herbal plant that increase the antibody. Do you transfer it, Dr. Tatang?
Yes, thank you. Ya, baik. Karena pertanyaan bahasa Indonesia, saya jawab bahasa Indonesia ya.
Iya, monggo.
Oke, baik. Terima kasih Bu Ida. Thank you for the question ya. Yes, right now we have a pandemic of COVID-19 ya. So we need to increase the antibody ya. Jadi meningkatkan antibody ya. So usually antibody can be produced by the presence of the stranger compound or stranger what is it? Stranger organism ya, come inside into our body ya. Maybe actually it's not about the antibody ya. Maybe for about the immunity ya, the immunity antibody also in the immunity, but immunity usually can produce by when for example the virus can come inside to our body, and then our body synthesize some compound that can be used by as antibody ya, as antibody. I think not for the antibody increase the antibody increase the antibody, but not a specific antibody for the specific virus I think but for example we use phenolic compound phenolic compound kind of the compound that can increase the antibody increase the antibody. So the tanaman herbal that have a phenolic compound inside can be increase the antibody. Yes, that's all.
Okay, the next question from Arif Ahmadi, Universitas Tadulako. As a person from small village in Indonesia, there are so many might develop in my village about healing potential for such an unknown leaves. Is it possible to use it widely, or should we make a research before we use it? Thank you very much.
Okay. Yes, for the safe, what they say, for the safe, yeah, for the safe, we need to do some research, yeah, about the first. The important thing is the toxicity, yeah. Maybe this leaf not have a bioactivity, but the toxicity, so make a dangerous, yeah, for our body, yeah. So we need to have some research before we use it, yeah, before use it, of course, yeah. But sometimes the person from the village, yeah, have something like ethnopharmacology, yeah, ethnopharmacology, that already try some sources from the plant that can be used by as medicine, yeah, as medicine, yeah. But for the unknown leaf, I think for the safety, we need to do the research first, yeah. Thank you, Mr. Arif Ahmadi from Tadulako. Okay, another question, Ms. Lina. Mrs. Lina.
There is several questions, Dr. Tatang.
Okay, in chat box, yeah.
Yes, in chat box. From Niputu Diantara, are there any conditions to produce a new natural product by bioconversion?
Which one? Are there any condition to produce a new natural product by bioconversion? Okay, because we have we use microorganism, yeah, microorganism. So we need to have a condition that switch comfort for the microorganism, yeah. For example, for the pH condition or temperature condition that will be mild, yeah. Condition will be mild, yeah. So we need to know first, yeah, about the condition of the microorganism growth, yeah. The condition of, for example, pH and temperature, the optimal temperature, yeah. And then from my colleagues, yeah, from Japan.
Yes, from
Mr.
Cahyo.
Mas Cahyo, ngopo tekon-tekon barang.
About lactone synthesis, Doctor. There any some reaction actually for hydroxylation? How to manage the hydroxylation? Is that radioselective?
Yeah, of course, yeah, hydroselective, yeah, because we use enzymatic reaction, yeah. Enzyme have only one reaction, or a specific reaction, yeah. So when we do the bioconversion, use microorganism or enzyme directly, yeah. So all the reaction in the lactone production, for example, for the hydroxylation, can manage the radioselectivity, yeah. Thank you, brother Nur Cahyo. The Gondrongers, yeah.
Yes. From the next question, Dr. Tatang.
Yes.
From Manarul Ijtihad, FMIPA Universitas Mataram. Some natural compounds are found in not halal organism. My question is, how about the use those material in our synthesis?
Okay. So, what is the not halal organism, yeah? Maybe the organism, ya, not the natural product, ya. Atau Pak Manarul bisa, Pak Istiad bisa menyampaikan, maksudnya ini natural produknya apa organismnya, Pak Manarul? The natural product, okay?
Mr. Manarul, you can.
The sources of the natural product or the organism?
Organism.
The organism. Thank you, Mr. Manarul. It's that, yeah. Okay. Some kind of the microorganism growth in not halal media, yeah, not halal media. So we need to choose that one, yeah, because when we produce by bioconversion, it will be impact to the halal status, yeah, the halal certification, because the organism has growth in the non-halal media. So we need to find or choose the organism that growth in halal media. Yeah, just like Saccharomyces can be what is it? Saccharomyces cerevisiae can be growth in non-halal media also. Yeah, just like in pig, what is it? Frog, yeah, in pork, yeah, pork lipid or another kind of pork, yeah.
Okay, the next question from Noraini Yunitasari. Izin bertanya kepada Dr. Tatang, untuk penelitian isolasi tanaman herbal, mohon masukkan perbandingan sampel dan pelarut yang optimal digunakan saat ekstraksi.
Oke, baik. Related to the solvent, ya, the solvent. Berarti bukan dia, Bu, ya. Ini cenderung kepada conventional extraction, Bu, ya, conventional extraction. Jadi perbandingan sampel dan pelarut. Nah, untuk ekstraksi banyak jenis ekstraksi, Bu, ya. Jadi ada ekstraksi yang apa? Maserasi, ya, ada yang menggunakan chocolate extraction, ya, ya. When we use chocolate extraction, we have only limited what is it? The solvent, ya, only limited solvent, because extraction of the chocolate is the continuous extraction, ya, the continuous extraction, ya. Jadi kalau pakai chocolate tentu saja kita hanya menggunakan pelarut yang lebih sedikit, ya. Tapi kalau kita menggunakan pelarut untuk misalnya metodenya adalah maserasi, ya, perendaman gitu, ya, maka pelarutnya tentu saja akan sangat banyak. Tapi kalau untuk menghindari pelarut yang kalau dalam maserasi yang faktor yang paling penting adalah dia terendam gitu, ya. Jadi semua sampel itu terendam, ya. Tapi kalau ingin mendapatkan supaya lebih ngirit gitu, ya, pelarutnya, maka setelah dilakukan perendaman atau maserasi, maka bisa difiltrasi atau didekantasi, ya, kita kemudian dapatkan kembali atau destilasi lagi pelarutnya untuk kita bisa gunakan kembali gitu. Itu, ya, Ibu Noraini Yunitasari from Gresik. Thank you.
Okay, thank you. The next question from our college, Habibie Hidayat, Dr. Habibie Hidayat. Thank you very much.
Okay.
Dr. Tatang, if we use anaerobic microbial such as Leuconostoc or Lactobacillus, how about the result of bioconversion in synthesis of natural product? Thank you.
Okay. Actually, Leuconostoc and Lactobacillus is the anaerobic microbial, ya. So the condition of the aerobic on anaerobic can be impact to the product, yeah. What is it? Facultative microbial, something like Saccharomyces cerevisiae, yeah. Saccharomyces cerevisiae, when in aerobic condition, we can produce bioethanol, but when we change into the anaerobic condition, the ethanol can be produced, yeah. Just like Leuconostoc and Lactobacillus. This is the also facultative microorganism, yeah. That's all, my colleagues, my brother Habibie Hidayat.
Yes, thank you, Dr. Tatang, for the nice presentation and inspiring. Okay, I'm sorry for Dr. Juliana. There is still a question for you. From Mrs. Febri Nitbani, Universitas Nusa Cendana. Dear Dr. Juliana, how about the toxicity of C8 compound? Do you transfer it?
Okay. Okay. Now, for the toxicity, we haven't tested for the toxicity. We just tested on the cytotoxicity. Is that answer the question? Because from the what we call that, from the relationship of the anti-cancer screening, we actually can also identify the cytotoxicity of the 4 compounds that already being sent for the test, I mean for the screening test. So for the cytotoxicity, we also determine by looking at the values of the percentage of viability and also the IC50 value. Meaning that for the cytotoxicity also, we can also determine by the IC50 value, and it shows that the IC50 value for the C8 gives the lowest compared to the 3 of the compounds. Does that answer the question?
Yes, doctor, thank you. And the last question from Mr. Arif.
Mrs. Oh, Mr.
Yes. Mr. Arif from ITS, I'm sorry. How about the purity of ligand and metal complexes? Did you purify them with simple column chromatography or by some advanced method? And the second question, how about the stability of ligand and complexes? Usually compounds with ketoenol tautomerization tend to degrade easily. And the third question, what is the acceptable value between theoretical and experimental value for elemental analysis? Is it okay to report for publication if the difference more than 15%? And the last, could you please describe the solubility of the ligand and metal complexes? In what solvent did they dissolve? Is it okay for the MTT assay? Thank you.
Okay, thank you for the question. Okay, the first question, okay, we did the simple column chromatography for the how to confirm the purity. Okay, just one simple column chromatography. And for the second one, about the stability. Okay, because regarding the stability, that's why the keto-enol tautomerization, okay, we know that tend to degrade easily. But okay, the keto-enol tautomerization, okay, in this case, the mechanism actually undergoes compound C1 with 6-hydroxy-4-methyl coumarin. That's why we don't even wait until for so long after we successfully synthesize C1, which actually undergoes the keto-enol tautomerization. Then we proceed to the next step where the azo coumarin. That's why it's straight away, meaning that we don't wait for a long time after we get the first C1, because the product of C1 is actually the one that undergoes the keto-enol tautomerization. So we go straight away, react with the azo coumarin derivative, and then from there we can further have some synthesis on what we call as the complexation. So that's why the main idea to prevent any degradation is to have this compound of C1 to have a straight away react with the azo coumarin. Okay, that will be the second answer. Okay, the third one, okay, what is the acceptable value between the theoretical and experimental value of elemental analysis? So this one will be very careful, because when you use CHN analysis, that will be called the difference of the analytical and also not the experimental and also the theoretical value. Okay, be careful, the difference cannot be more than 50%. Normally it's about acceptable is about the most is 10%. So if let's say we have more than 50%, so we have to repeat, try to send it again to have the second test on the CHN analysis. And then for the fourth question, the solubility of the ligand and the metal complexes. Okay. In order for us to send the compound, first is for the NMR analysis because we have this in liquid form. So we try to have some kind of testing regarding the solvent that can be used to dissolve totally with the product, and it shows that the best will be the MSO. And we can say that luckily the MSO is the best solvent to dissolve with the compounds, and it does not affect the MTT assay because for the MTT assay, usually we don't use the same compound where we have to react for the NMR. So for the MCSA, I was also told that using the same type of solvent, which is the MSO. Okay, so I think that's the answer for all the 4 questions. Is that okay? Is that clear?
Okay, it's clear. Thank you, Dr. Juliana, for the answer. I think Q&A session is enough. The speakers, the host, and all of the participants. From the discussion today, we can conclude that organic chemistry promising for future life, and absolutely is it not scary or difficult because organic compound essential to human functioning. It's like carbohydrate, lipid, protein, etc. So the meme that I shared in the beginning of this webinar just a joke. Thank you, Professor Madya Dr. Juliana Binti Jumal and Dr. Tatang Sabur Julianto. And thank you everyone for attending this webinar, and have a great rest of your day. I return the screen to Professor Is Fatima.
Thank you very much, Ms. Lina, Dr. Juliana, and Dr. Tatang Sabur Julianto. I'm sure that discussing chemistry from living organism as well as exploration from plant such as bioconversion is very interesting and always interesting. So that's why discussing organic chemistry, especially from biodiversity, is always the hot topics. That's why there are so many questions from the participants who are very interested to your inspiring talk from both of you. So as appreciation from our department, we'll give the certificate to Prof. Madya Dr. Juliana Binti Jumal for a very nice talk. Also to Dr. Tatang Sabur Julianto for very inspiring talk, and still keep spirit for the next research, Pak Tatang, for our department and also for Indonesia and also to the world. And also we thanks to Miss Lina for very nice moderating the session. Thank you very much, Mbak Lina. And for all participants, the certificate can be downloaded by this link. I will share the link into the chat room just a moment. And for information, because the webinar is part of the Global Engagement Grant in collaboration with World Class Professor Program. So this is the 6th webinar series of our department. And the 7th webinar series will be conducted in 15 October 2020 with the theme of chemical education, being inspiring teacher for future generation, will be conducted starting from 9 until 12 with Prof. Johan Yoga, an international mind map trainer for Asia with affiliation in Singapore. So don't miss the next agenda. And thank you very much for attending this webinar today. And good luck for all of you Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam
Thank you,
warahmatullahi
Dr.
wabarakatuh.
Juliana.
Thank you very much, Prof,
Yeah.
and everyone there for inviting me. Thank you for the
Hope to see you
sharing.
again.
Inshallah, we'll see
Yeah.
you next time.
Yeah, hopefully the pandemic recover and we can meet physically. We
Yes,
invite you to our
hopefully.
department.
Yeah, hope to see you again next time.
Yeah.
Okay,
Thank you to everyone.